topics metabarcoding as a promising conservation tool to monitor fish diversity in Indonesia marine protected areas. We are thrilled to have you join us as we delve into one of the most innovative and impactful approaches in marine biodiversity conservation. I would like to thank you, our Dean, Bapak Beginner Subhan, that already joined us, and our embryo director, Ibu Dea Fauzia Lestari, and of course our uh, presenter today, uh, Ibu. I usually call her Mbak, but at this moment, in this formal event, I will call her Bu Dita Cahyani. Welcome. And today, before we going into our talks, first I would like to invite Ibu Dea Fauzia Lestari to give her opening. Bu Dea, the screen and time is yours. Thank you.
Thank you, Bu Mutia. Also, I always call Mbak Mutia, but in this occasion, I will call you Bu, also like Budita. Okay, thank you very much. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Uh, Thank you everyone for coming, and also we have our Dean, new Dean, Pak Begin. Thank you for coming. Yeah, Embryo is a think tank organization in Faculty of Fisheries, Marine Science and Marine Sciences, IPB University, and we are focused on the research of marine biodiversity, especially. But yeah, here we also have embryo member ya, Mas Gusti, and also the others. I also see here Sebastian and the others maybe from another institution. Thank you for coming, and I hope from this Embryo Talk series we can collaborate and also have a new idea and this good discussion to enhance the research, especially for marine biodiversity. And once again, thank you Mbak Dita, Bu Dita, for your availability to give the talk today. And maybe I will not give long speech or talk because I believe we will have a very nice research or information from Budita, and then I will give it back to Bu Mutia.
Okay, thank you very much, Bu Dea, for the opening information. Then, if not speech, then I would like to invite our new dean, Bapak Beginner Subhan, to give him his opening speech. Pak Din jadinya,
Pak
Pak Dekan.
Din.
Screen and time is yours, please. Thank you.
Yeah, thank you very much. Good afternoon, ladies and gentlemen. Thank you very much. First of all, congratulations yeah, for Embryo starting our first Embryo talk yeah, in 2026. And thank you very much for organize still consistent and persistent yeah, to make embryo as a hub for the knowledge exchange yeah, for anything yeah, related to the biology, fisheries, and what we call in IPB agro-maritime. And thank you for Ibu Dita, Mbak Dita Cahyani, ya. Ini our beloved friends who always support us, ya. And then, and I think the topic is very important for us, ya. eDNA, and it will be very good sharing, ya, about eDNA and give us hopefully give us insight what happened now related to eDNA research, yeah, in Indonesia or also in the world. And I also want to say thank you for the all audience, yeah, Embryo Talk, and we hope that this talk will be good time for sharing, yeah, sharing knowledge, information, and after that we can collaborate, yeah, in the next probably research or teaching or etc. I think that's all. Enjoy the talk and yeah, happy Ramadan. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Waalaikumsalam warahmatullahi
wabarakatuh. Thank you, Pak Begin, for the very welcoming speech. Then today's session, I would like to make an introduction again. Will be led by our distinguished speaker, Ibu Ni Kadek Dita Cahyani, Ph.D. from the Department of Biology, Faculty of Science and Mathematics at Diponegoro University. Her expertise in marine biology and her pioneering work in eDNA, especially and metabarcoding, has contributed significantly to understanding of fish diversity in marine protected areas. Without further ado, Mbak Dita, Bu Dita, the time and screen is yours, please. Thank you.
Thank you, Mbak Mutia, Bang Begin, and also Dea. I just realized that I have to talk in English, so hopefully, I thought it would be like in Bahasa, but hopefully it's not, you know. So wait, let me share my screen. Okay. So first of all, I just want to say thank you so much for the invitation. Thank you, Dea, for inviting me. And this is the first talk for the embryo on this year. So I'm very honored. So today, we gonna talk a bit about eDNA. I know that the title is actually related with our new paper that coming this year, but I just want to give you some idea about how this eDNA become one of the leading research in Indonesia and in the world, and how can we work together moving forward with this method. So my name is Dita Cahyani. Now I'm currently lecturer at the Biology Department, Faculty of Science and Mathematics, Universitas Diponegoro. I've been working with Bang Begin for too long. It's like more than 10 years, I think. For the first time, we're doing molecular and there's so many, like projects that we've been working with, and it's a pleasure because then we are here today with the newest technology that we can use to study the biodiversity, especially marine biodiversity. So the first thing that I just want to remind you that why eDNA, environmental DNA, become one of the leading method is because everything, the living organism, all the living organism, from the microorganism to the metazoan, has DNA in their cells. And the fact that this DNA or this cell is not only present inside the organism, but also they being what you call it, they being released into the environment. That's the source of the DNA in the environmental DNA. So environmental DNA, as the word, is actually the genetic material that being shared by the organism and live into the environment. So we can find the eDNA in water and then soil, even air. and also feces, mucus. So there are so many or a lot of sources of the eDNA. As a methodology, eDNA is actually a high sensitive and non-invasive technology that we can use using molecular technique to identify species or to detect the species. So this is kind of like a powerful method that we use, and it is helping a lot of ecologists, especially to assess the biodiversity. The application of environmental DNA is actually very, very wide currently. So the first is how we can detect the invasive species, for example, that coming into the environment, and then how we can see the impact of the pollution industry into the environment. And I actually work on, we start working with the pollen biodiversity that we collect from the bees to get the plant biodiversity. So we collect all the pollens, we do the metabarcoding, and then we try to find where the pollen came from, because this is the source of, for example, for honey production. So we can understand how the environment affecting the quality and the quantity of the honey in a certain places. So the difference between eDNA and the Sanger method that we usually use is because we use the high throughput sequencing. So this is like when we're working on the eDNA, there's a lot of new term that we have to understand. The first is the high throughput sequencing. So this method that being used in the eDNA first is because our target is not directly observable in the environment. So we use eDNA because we don't, sometimes we cannot see the organism, but we know that there will be the DNA in the environment. The second is the DNA, because we call this is being shared by the organism. The quantity of the eDNA, they will be very low, and the quality is also very low. So because of that, we need some technology to be able to amplify the DNA and read them in the low quantity. And then the third one is because we need the method to detect multiple species or genetic markers simultaneously in a one run. So instead of working with one individual, we're working with the eDNA in the samples, and then we want to detect multiple species that might be present in the environment. So that's the idea of the eDNA. And then there's also a lot of discussion, like a hot discussion about which one is the correct term when we use the eDNA, what type of samples that we have to collect for this. There usually two terms. So if you talk about the metabarcoding or the barcoding, molecular barcoding, that's the approach that we use to study the diversity. But they also like the samples type. If you collect the samples from a bulk sample, for example, you're towing your plankton, you will get the plankton, and then everything inside those, there will be like high quality and high quantity of DNA. So that type of sample we call bulk sample. However, if you collect the samples from the DNA that being released by organism, not directly the organism, then you will get a very low quantity and low quality. So that's exactly what we call eDNA. However, currently there's an interchangeable of using both of these contexts, both of these terms. If you use eDNA or the molecular as a whole, there's a two approach. The first is you are doing what we call amplicon sequencing, where you target one single gene or amplicon, or you just do a shotgun, which is you You basically get all of the genome in those organisms, and then you align them at the end by bioinformatics. And the approach that I use mostly and I will introduce today is metabarcoding. So I targeting one gene, especially CO1, for marine organism, and then I will try to identify everything in the samples based on the gene, based on the CO1 gene. There also another approach in the eDNA. I'm not really using this method a lot, which is the qPCR. So this is basically just like what happened during COVID, where we targeted a specific organism, and then we quantify how many replicates that happen in the PCR. So the qPCR is usually using in the eDNA is usually using to get like a rare species or the introduced species, where we want to know whether they're being found in the environment or not. So there's a present absent type of thing. Meanwhile, the DNA metabarcoding that we will talk today is actually you identify a short sequence in your samples that being amplified to every organism that has the gene, and then you identify each of them. So in the one sample, you will have like a 100 or million of sequence, and then you will identify a couple tens of 100 of species out of that data. So that's the term that we use today. And the other thing that this is usually, I know that if some of you already work with eDNA, work with the NGS, I've been challenged by a lot of people about why you're using the term metagenomic, why you using the term of microbiome. Basically, both all of the term has their own reason to be used. For example, metagenomic. Metagenomic is the study of material or genetic from the environmental sample. So everything is metagenomic, but which approach that you use? Are you using the metabarcoding or are you using the qPCR? That's another thing that follow up this term that you use. Metagenomic also meaning that you try to find the function, the diversity of the function of the organism, especially microorganisms. So if you want to see, like, for example, in soil, you have the bacteria that were that doing more on the one metabolism, then the metagenomic is actually your approach. So this is like the term that always give me a hard time whenever I have to explain what is eDNA, what is metagenomic. People like to challenge us, then sometimes to just learn something new. And microbiome at the end is actually the collection of microorganisms, and then you can detect the microbiome using metagenomic, or you can detect the microbiome using metabarcoding. So this is the difference. The other thing that really helping this method to be developed is actually the sequencing platform. The first we use the Sanger sequencing, and then the second generation sequencing, which is Illumina coming, and then the third generation sequencing, which is one of them is Oxford Nanopore, is also being using a lot. So Illumina is one of the best currently because it read a short sequence. It reads a short sequence because they want to make sure that they do a good reading. So the validity or the quality of the reading is really high. However, the Oxford Nanopore, the third generation, try to read longer sequence. Sometimes they want to read the whole molecules of DNA, but the quality on how they read each of those nucleotide is less than the Illumina. So both of them is actually having their pros and cons, but of course, some of them is just depend on how we as a researchers want to apply them on our research. So before I give you the example on how or what we can work with the eDNA, I just want to give you to list some of the challenges on eDNA research. The first is, of course, the molecular method. So some of the people don't want to just jump from being an ecologist and then being a molecular researchers. So molecular method here is really challenging because it need time and skills for people to learn about it. The second is bioinformatics, the data that you get from the eDNA is actually very big data. So you have to equip yourself with the bioinformatics skill to do the analysis. And then the funding and the cost. The cost is still higher than the other method. For example, if we do the visual census. So this is also one of the challenges. And of course, the last one is the technical or the sampling method, contamination, statistical validation. This is the thing that people get very scared if, when they're being, for example, like me, I've been taught by a group of people from the US where they have like a strict restriction about how the contamination have to be contained. However, when we work to Indonesia, we have to adjust. We don't have all the high tech facility in the lab, so we have to adjust with what we have. So for me, this is just the level of how we gonna use this method in the field. It doesn't have to be very strict based on what other people taught us. So I just want to make sure that all of you not being scared by people if they say like, oh, you have to do some, you have to get a good statistical validation, for example, so you have to get less minimum, for example, 10 samples. Meanwhile, all we have the funding is only to work on 3 samples, for example. But that's the challenges. And I'm here to discuss how we can move forward with these challenges so it's not hinder us to work on the eDNA in the future. So this is related with what we have done with the project that we just published. So basically, we just want to test a method. Actually, that's the idea at first, because we know that when you want to see the eDNA in the water, you can use many methods. You can use Niskin, you can use the intake water, you can use the bucket, or you can just use like a Fundorn, for example, and you still get a good result. So for us, we test 2 methods, and we end up with using the second one, the one in the right. The one in the left is actually the one that we use, the vacuum pump. And then the one in the left is the one that not using electricity. We use like an IV bag, and then put everything in the sterific. So this is actually the paper, what do you call it? The one that we apa namanya, filter paper. So this is the filter paper, not like the one that we use in the vacuum pump, but inside the sterific. The project that we got is from University of Washington, Seattle, under the eDNA collaborative project. So basically, we want to test how eDNA can be applied in the most biodiverse country, but at the same time, we have a very low in the skills, and also we have no experience in bioinformatics before, and a low budget for the project like this. So we choose the one without the electricity sampling. And then we also use the ONT, the 3rd generation sequencing, because it's more handy. And since I'm in the university, it's really easy to use as an education for the student. So there's a 2 paper coming from this project. You can just download this paper, but I will not talk about this. I just want to show you how we do it, and how everybody can also repeating the same thing that we propose in this paper. So we compare the data with the visual census method. The problem is the visual census data that we got is actually from the 3 or 4 years before we sampling the eDNA. So there's a discrepancy a bit, but actually overall we can compare why or what we get from this paper. So we being inspired by this project that being done in Florida, United States, where they can bring their R&D and their their lab equipment into the field. We're not really doing like this, but at least this is the inspiration of our projects. We started project on 2022. And then we sampling the sample in the 4 different zonation in the Karimun Jawa National Park. And this is what we do. So we dive in, get 1 liter of samples, and then do the filtration. The data is basically, there's nothing really fancy about the data. The data is just the data. We got several species that we can see in every location we sampling. There's no significantly different in the alpha diversity, but there is a higher in the tourism area, in the open access. But and then we can see whether which species is being shared from the eDNA and visual census. But the thing is, I think this is the most important thing that we want to highlight, that the data that we found, the species that we found using the eDNA is actually related with the nocturnal samples, the nocturnal fishes, Amphidromus, Oceanodromus, and also pelagic and eritic. However, the one that we usually get from the visual census is more reef associate and non-migratory species. So the take-home message was that if you want to get the data for nocturnal fishes, Amphidromus fishes, demersal, eDNA is really helpful on this, because that's some of the species that you cannot get during visual census, because usually we do the visual census in the daytime. The other one that really excited is we learned the whole new bioinformatic analysis. This is really fun, but it's challenging experience. But we finally make one simple analysis that we can share, and then we also try to expand this research into another one. So this is the eDNA that you guys can apply. You can use for aquaculture when you try to find, for example, the gut content for shrimp, for fishes, not only the microbiome, but also the food content. You can do the biodiversity assessment for so many different ecosystems. You can also try to see whether using a different type of, using different gene target, can you get some of the rare species, for example. And then this is some of the project that been done outside Indonesia that I put it here just to, give you an idea on how we can work moving forward with this. You can see the invasive species, and this is the ballast water. I think they are working on the ballast water as well. But biofouling, this is also a nice thing to do using the eDNA. But this is the cetacea, but this is the thing that I want to do next. So I really want to use eDNA to see the blue carbon, the organic carbon from the seagrass, or also from the mangrove. There's a couple of people working on this currently, but I know that if we want to start this, we need a good team that working not only doing the eDNA, but also the ecology of this ecosystem. But this is the thing that I want to propose to everybody here. Maybe we can start working on this project, because then we can leverage the eDNA to answer many other questions. I think that's all what I want to share today. I think it's good to have a discussion on how we can work on this. And again, I would like to say thank you to everybody. And yeah, let's have a good discussion. Thank you.
Okay, thank you very much, Mbak Dita, for a very inspiring and interesting talks. Me myself have several questions, but we could hold that first because we still have many times ahead of us. For now, I would like to invite our participant if there any of you have questions or discussion or even a collaboration proposal to Budita. Please raise your hand and then you could introduce yourself. We already have 77 participants at the moment, so it's quite merrier here, but still a little silence. Okay, is there any questions, or if you feel a bit not in mood talking in English, you could talk in Bahasa. You are very welcome. Okay.
But
So
I think Sebastian have a question.
okay. Please, Sebastian.
Hello, selamat sore, and many
Selamat
thanks for the
sore.
very interesting presentation. It's good to see you. I was very interested in your explanation of the different methods because I'm not an expert in eDNA, but I'm about to start on a large project where we include eDNA as one of the methodologies. So you were mentioning, I think, for qPCR that it's possible to also detect abundances of individual species. So one of the things that we're planning to do in our project is to reconstruct food webs and abundances of organisms based on eDNA. And I would like to know a little bit more from you what you think about that approach and how it's possible to detect not only presence, absence, but also abundance, or maybe I misunderstood. Terima kasih.
Okay, thank you, Sebastian, for very interesting questions, Mbak Dita.
Okay. Thank you, Sebastian. First of all, this is the one challenging question in eDNA. I know that we cannot decide what you can extrapolate the abundance of the reeds to be abundant of species, right? But I know there's a couple of group or couple of teams currently try to move forward with that. So they want to get some idea if we get this type of abundant of read sequence, they can what you call it, extrapolate it to the abundance of the species. But again, this is still a debatable because theoretically that's not how we count the species. But for qPCR, I think what I mean is that you just want to see whether you get more DNA of specific organism in a location. But I'm not sure if we can directly say that the abundance of those species, because again, the concept of eDNA, if you guys remember, if you see like a school of small fishes, you will get a lot of DNA, right? But at the same time, if you have one individual of a whale, for example, you still get a lot of DNA from those, but you cannot just extrapolate them based on the number of the DNA. So for me, I don't think we can use it as the real abundant, but that's not what you call it. I think in the future there will be some statistical way to do that, because I saw a couple of, I think it's still in the project proposal on how they want to count them as, I don't know how they do that, but yes, for qPCR, the thing that we can decide is only like which one is bigger in number, but not the exact number. I think that's the one that I can answer for now.
Okay, thanks a lot. And with regards to the blue carbon sources, that's also very interesting. I'll send you an email about that because there might be an opportunity to
Thank
collaborate.
you. Of course. Of course. Thank you so much, Sebastian.
Thank you.
Okay, thank you. I think the question is answered very well. Actually, that is one of my questions to Mbak Dita because I have someone from Brin Laterio told me to do that, but I still figuring how to exactly quantify the DNA into species because it's like you said before. Okay, perhaps we have another participants.
Yeah, there's one, I think,
Yeah.
raising.
So please, Pak Wasgiono, perhaps you could introduce yourself from what institution and your institution.
Thank you, Bu Mutia and Bu Dita, for nice presentation and arrangement for today's meeting. My name is Arif Wujdi, not Waskiono actually. Currently, I'm a Ph.D. student in Pukyong National University, South Korea. And I'm currently doing with the eDNA as well for the fish community and also for the stony coral in one of high use MPA in Nusa Penida with the local NGO there. And I also have the question continuing with the Sebastian question. Beside of the abundance, I also wondering when the eDNA or the genetic material also come from the dead organism, right? Because that organism also still releasing the genetic material in the environment. But the main question is how we can measure the dead organism and then how to follow up when we find the DNA, like for example, from the dead whale, the organism is only one, but the genetic material is so many released to the environment. But in fact, the whale is dead, not living anymore. And in terms of the species distribution and occurrence, we cannot say that the whale is inhabit in that area, isn't it? Yeah. So how to follow up for this case? Thank you.
Okay, thank you. This is a very interesting questions because
It is.
many of our
Yes.
colleagues, I think, have the same problem with that concerning ecology. So please, Mbak Dita.
Yeah, so this is a good question. It's really hard to answer. But I
Yeah.
just want to be I just want to be um we cannot know which one is alive and which one is not, because if we check them from the quality and the quantity of DNA, they will be the same. If the quantity or the quality is getting lower, there's not only because they're already dead, but they might be because they're already being too long in the environment. So, so my answer is we cannot see whether the species being dead or not from their only from their the eDNA. However, at the same time, eDNA is only a complementary method. It cannot be used solely only eDNA, because then we have to have the other method to see the ecology, the biology of the location. For example, we try to get the eDNA from the deep sea in Alor for the extreme upwelling. We do get the species from the deep sea, but it's not directly said it is coming, the upwelling is coming from those depths. So there's also have to be another study on the oceanography that support that. But that's a fact. There's a DNA in the 1,000 meters species going into the upwelling water. But again, it's not, we cannot just conclude that they are coming from that location. So this is the for me, this is the challenge, but also the fun games on the eDNA, because then you can ask a lot of questions, but you also need to add more data on this. So for example, like the whale example, if we have the data from oceanography, from maybe people telling us there's a whale died in some of the places, for example, then we can conclude where the eDNA coming from. But if not, there's just an eDNA present around those places. We don't really know whether they are live or not. So I think that's how we can conclude. So some of the question is cannot directly answer by eDNA, I think, especially for this type of example, though. I think that's but yeah.
Yes, thank you. Thank you, Bu Dita. Yeah.
Okay, thank you, Mbak Dita, for the nice answer. And well, actually, I have a result that came from the upwelling area, and we are talking about not exactly about eDNA, but rather I would like to call it metabarcoding, because we are doing some plankton
I see.
using waters that taken with skin and then filtering it. And however, from the same column depth, and using the same skin, and we make it into two bottles, we have a very different result, whether it's percent coverage and the composition. So yeah, I think there is. That's why I very agree with your comment about this is a complementary, or like a monitoring. So we need to add more data to confirm our the eDNA analysis results, I think, like that. So if we have any other questions from participants? So, Mbak Dita, if I could ask, is there any threshold? Because sometimes when we are working with eDNA, we are like tempted to say, so we have this species and this species and this species, using these very short sequences, and then using just one, no repetition of samples. So is there any threshold for the percent coverage? Because when we are running our samples and then we have this percent similarity, I usually told my students, if you have uh, less than 95% or 97%, you could not talk on species. I say that, but is there any threshold on that?
Yeah, it's different though. In for fish, we also using the 95%, 97 to 95. For 18S, protist single cell eukaryote, or usually 18S also collect plankton. We usually use 98 to 99% similarity. But if you work on microbes, we use 99%. That's the first threshold to get the OTU. But at the same time, we also do some cleanup during bioinformatics. For example, we not retain the singleton and doubleton. So we delete all the singleton, we delete all the doubleton. And then next, if we work with fish, I usually work again with R fish base. So we compare our data to fish base and make sure that all the species that we being identified only present in Indonesia or Asia Pacific, like based on our location. So we delete everything that the distribution outside Indonesia. So there's a couple of different layer on clean up the data. Usually that's how we do, but for the threshold on the similarity, we also use 95%. Well, usually we play first on the data, and the best one for now, we're using the 12S for fish is 97. Is it 97? 97% of similarity. But again, before that, we clean up so many things. So we delete all the singleton, and then we delete, usually people usually said only singleton, but we also delete doubleton. So if you have a sequence with only two abundance in the samples, we just delete them. So that's how the layering analysis that we usually do. But yeah, it's so this is the thing. We come up with 95, and then the reviewer is actually telling us to do the 97. We changed to 97. Some of them say, what about you doing again, like make it more stringent, like 98. So we end up doing all the analysis and give them the result. The best one is 97. So this is, if you ask me like what's the baseline, like where it come from, it's actually because the reviewer is just really they just like they keep asking us to try things, because that meaning that there's no a good or one what do you call it, an exact threshold for this. So as long as we can what you call it, defense to them about what our data, they usually fall by 95 to 97 or 98 similarity. Yeah.
Okay, Dita, that deleting the singleton and doubleton is also interesting, because we have more accurate data, in my opinion. However, sometimes I found people that are working and claiming there is invasive species using that data. And, so
Exactly.
how continuing to that statement, how we could determine if in our samples we are finding an invasive species. Is there any category, or perhaps the number of OTUs that founding in that results, or how?
Yeah, well, that's a really interesting question, Mbak Mutia, because that's what happened with our freshwater sample, freshwater fish. We do all the cleanup, singleton, doubleton, and then only Indonesian. But at the same time, there's a couple of species that abundance, there's quite abundance, but not from Indonesia. So when we see the data, we decide not deleting them, so we include them. But I don't have like a specific number, like how many, like the abundance should be more than 10% or 5%. I don't have it. But as long as they still fall into not a singleton, for example, and they present more than once in the replicate, we usually retain the data and then we come back to the story, like behind about the biology and ecology of the places, because we work in the lake, in the like a closed environment. We can get a better understanding about what might be released in those locations. So that's how I like back and forward with the comparison, but we don't have any specific number. So that's why I'm saying I always being challenged by a lot of people. Although presenting in the conference, some people will ask me like, how can you be sure? And my answer is always, I'm not sure, but this is the result. So I only presenting the result and how I what you call it I'm discussing this result. It's not there's nothing like very sure that invasive species is there. But usually, if we get a lot of, for example, the red devil fish in some of the lakes, we do have a lot of like the abundance is quite high, so we can see that directly. But I don't have any specific number. As long as they fall after, I mean, in the data after we clean up, we are our argument is that they might be there. But but if we're not getting the data from the fishermen or not, then we can say there's a possibility of there's a invasive species. So like using the wording on the report is also important. So it's not like we are very sure, 100% sure, but there's a possibility. So yeah, something like that we're still working on. But yeah, that's always happened.
But it's nice to hear it from your experience, because then we would be more aware of that. Perhaps some
True.
of our participants are doubting his or her result,
Yeah.
founding a very different species, but in singleton or doubleton, is it really invasive or not? So yeah.
Yeah, true. But so Mbak I remember one thing. We also find out the contamination in our lab based on this the way we clean up the data. So when we work with the marine fish at first, and then we go to the freshwater. But then like when we work on the second sample on the freshwater, we just realized that we get more marine feces. So the first is we said like, oh, there may be the bioinformatic, there might be because they sort, they cannot distinguish the fresh water and the marine water. But then we try to do, like I said, changing the threshold, like 90%, 95%. And we just realized that is contamination, because then the fish, the marine, the freshwater fish in different location have a different pattern. Like in Lake One, they have some of the species is abundant compared to the other. The other location have a different pattern. But the number of the marine fishes and the species is the same. So that's make us realize that there might be a contamination in our laboratory work. So that's why there's also another thing that when you have the data, you have very careful on reading your data and comparing it with the what you call it with the journals or with the research that people have. So that's what happened. Yeah. Halo, Mbak Mutia? Are we losing Mbak Mutia? Oh, okay.
Yeah, I was thrown from the Zoom. Yeah, it is very interesting from your experience and your talk today. But unfortunately, we have a very limited time. So uh, perhaps I would like to ask if there are any one questions from the participants. Okay, so far we still haven't. Perhaps they still uh syncing the informations. First, Mbak Dita, it is very insightful and interesting talks we have and discussion we have today. And about the blue carbon, it is also very interesting because I have worked with
Yes.
microbial in seagrass ecosystem,
Mhm.
and right now our team is working with the blue carbon in seagrass in marine lakes, and we are trying
Mm.
to the micro organisms layering in the sediments and the root rhizosphere and the leaves area. So perhaps we could talk in other time about that and collaborate. Perhaps I very hopeful that we could arrange something in the future, perhaps. And probably if there is any questions from the participants, is if they would like to contact you, is it allowed, or
Yes,
they
definitely. Just of course,
yes?
yeah, of course. You can just contact, well, Dea Bambegin knows my phone number, so you can just WhatsApp me or just send an email if you need like a longer answer, or just like just contact me. Maybe some of you need some discussion. There's also we can do that as well. And yes, Mbak Mutia, for a project, I'm really interested with that because it's a challenge doing something new, right? So Widya also said something, but yeah, because the eDNA is for me it's just like they still develop and there's so many questions that we can actually answer by this method. Yeah. I think I put my email on the last slide and then I can share it the slide to Adea as well.
Yeah, and also the committee will share the video on YouTube channel of our faculty. So someone who missed some
Oh.
part of the presentation also can still get the information from the video. So visit our YouTube. And also the certificate, the committee will share to your email. So please fill the attendance form, and the committee already put in the meeting chat. So don't forget to give your information about your email and your name.
Okay, thank you very much, Mbak Dia, for the information to be noticed by the participants. However, we already reached 3 p.m. So a very nice afternoon and ngabuburit with you, Mbak Dita, and our all participants. I would very like to thank you for today talks, and to our embryo team. Our director Budaya, I think he has another agenda. He's as busy as a dean can be. So thank you very much, everyone. Thank you, Mbak Dita, for the interesting talk, and
Thank you
thank
so
you.
much.
So
Thank
I
you,
will
Mbak
take
Mutia.
back to Mbak Dea, perhaps. Thank you.
Yeah, see you in the next Embryo Talk series. I think Mas Ijat already agreed for the next talk series and 10 of March. So see you in next ATS.
Thank you
Okay,
very
thank
much. Have
you.
a
Thank you,
nice
everyone.
recording. Thank you.
Makasih
Makasih,
semuanya.
Dita. Ntar
Sampai
kontak-kontakan
ketemu
ya, Mbak.
lagi.
Mbak
Iya,
Dita,
iya
terima
pasti.
kasih
Sama-sama,
everyone.
Dea. Thank you, thank you.
Ada
Makasih semuanya.
Mbak Citra.